Okay, so hey, I'm Tyler. I'm from Western University up in Canada, and I'm going to be talking to you today about transfer learning for cross species bacterial sgRNA Cas9 activity prediction. So, to begin, we have a nice cartoon of Cas9, and Cas9 is localized to double strand DNA by a guide sequence, a single guide RNA. And it uses that guide RNA alongside the PAM. In this case, for what we're looking at using SP-Cas9, that's an NGG PAM. So when you have a specific target site like this, question would be, is this a good target to choose from? And moreover, just to highlight things here, there's a lot of options available for places that you can target. There's one target for every 8 nucleotides. So any instance of a GG or, of course, CC with the other strand then being an optional target. That's a lot of different target sites to parse through. And as much as this may seem like a nice sandbox of choose your own place to target with Cas9, unfortunately, on-target activity varies. Even though we like to believe that we just choose something, have a perfect match with the guide, have that presence of a PAM, and it's going to be highly active, it is unfortunately not the case. And we can see this across these 4 different data sets in different bacteria. So to solve this, we have a nice simple process here using machine learning. So we take as an input the Cas9 target site. In fact, we take a lot of these target sites as input. Now, this is just the nucleotides. It's then encoded using one-hot encoding into a nice binary matrix. It's just 4 by n, with n being the length of the input sequence and 4 just being those 4 nucleotide options. We then pass those one-hot encoded target sites into the model. And this is just an initial model architecture. It is updated a little bit. But overall, it starts with a convolutional neural network layer. Just the idea there is to capture local adjacent nucleotide impacts. It's then passed to a recurrent neural network, specifically a bidirectional gated recurrent unit. And the goal there is to try to identify more sequential features. Then it's just passed to a dense neural network, and we get an output prediction. Now, of course, when you have an output prediction, if you want to train a model, you need to use some sort of error metric, and therefore you need to have a score. So that brings me to our E. coli enrichment system. Prior bacterial work has generated some nice data sets using a method called killing efficiency. In bacteria, if you target a bacterial genome with CRISPR-Cas9 and it cleaves, then it will kill the bacteria generally. Unfortunately, if you chat with your microbiologist friends, you'll find that bacteria also just kind of die. So it's not always the best metric when you're trying to look at performance. So here we have an enrichment system. Rather than targeting the genome, what we have is 2 plasmids. One of the plasmids harbors the Cas9 and the guide sequence, and it's targeted to another plasmid, which encodes a toxin, a CCDB toxin, which just prevents bacterial growth. So therefore, if you have a good sgRNA, then you will get Cas9 cleavage of that toxic plasmid, and therefore you'll see bacterial growth. And we can measure this using read counts in the before and after. Now, as much as this is great, and we believe our data set to be nice and perfect, it's unfortunately pretty small. We have 2 different versions, the same guides, but just 2 different nucleases, TEV-SP-Cas9 and SP-Cas9. Unfortunately, there are about 300 guides, which is not really great for training a robust model. Conversely, there does exist those pre-existing killing efficiency data sets, and those are quite large at about 45,000 guides. So to take advantage of this, we've used the concept of transfer learning. So now present kind of an updated model architecture. On the left side, you can see the same path that I showed before, just the convolutional neural network into an RNN and dense layer. But there's also, as we call it, a dual branch architecture. The other side has several layers of CNNs, which we found to be most conducive to training with the larger pre-existing data set. So the idea take that pre-existing killing efficiency data set, train up the whole model, and then take what I refer to as this base model, freeze most of the layers within it, and then train up the model with our data, creating two different versions of the model for the two different nucleases, TEV-SP-Cas9 and SP-Cas9. So how does this work in terms of performance? Well, what you can see here is the 5-fold cross-validation performance for both the TEV-SP-Cas9 and SP-Cas9 versions of the model. And we're pretty happy with this performance. We're using a Spearman rank correlation coefficient metric, and we're getting about 0.63, which is significantly better than pre-existing models. Now, something to note is that earlier on I mentioned that the input is 4 by n. And I didn't specify the normal guide sequence length or the target site length of 20 nucleotides because we tested some things up and downstream. What you can see in the middle of each of these plots is that little rectangle. That's just representing the sgRNA target site itself. And then incrementally, we're adding on a nucleotide moving either upstream or downstream. And what we found is that although upstream just seems to contain a bunch of noise, downstream, we found some improvement to model performance from adding on additional nucleotides. And this was something that was really nice to see because recently there's been some research looking at something called a post-PAM interaction between Cas9 nucleases and the target site itself. So it was really good to have this recapitulation between model performance improving when seeing those nucleotides, and then also having that biological research, which shows that there are actual contacts happening there. Moving back to the 5-fold cross-validation performance that I showed, a lot of people are probably thinking, great, yeah, you showed 5-fold cross-validation. That's not much of a test. That's a good initial stage. And I would agree. Thankfully, we had the option of using 3 different data sets to test generalization performance with. So the first one here was a pre-existing killing efficiency data set generated in E. coli. And this was a nice test of model performance for 3 main reasons. For one thing, we have differences in enzymes, but I think more importantly, it's a killing efficiency data set, not enrichment, and it was generated by someone else using a completely different activity score metric. And we still found performance results of about 0.68 and 0.66. But then I move on to the bigger finding. We ended up generating a data set in a completely different species. E. coli is nice, but there are other bacteria that are available. And the thing that we were really happy to find is that the model generalizes across bacterial species. And with results of about 0.64, 0.61, depending on the 2 versions of the model. And this was really great because if you take a look at any eukaryotic Cas9 prediction models, what you'll find is that they cannot generalize between species. And then if you get a little more granular and look at the specific cell lines, they can't move between a hex cell and a HeLa cell. So it was really nice to find that in bacteria, we actually get this generalization capability. Finally, we also just tested things with a small fragment from Salmonella enterica, and this was just a nice final test. It was done on a fragment from Salmonella enterica in E. coli, so it was kind of a pseudo cross-species test. But once again, we have pretty good performance results here. Finally, just a nice little thing. These models are now available online, so I don't have to get that annoyed look from a microbiologist when I tell them to install a Conda environment. But yeah, overall, what I've shown is that through the use of kind of a novel architecture and the concept of transfer learning, we can take these small data sets and generate models with high performance that are capable of moving across bacterial species. And once again, the model is available online. Just want to give a couple acknowledgements to Doctors Gregory Glure, Dave Edgell, Mike Hallett, as well as Dalton Ham, who generated most of the data used in this presentation. as well as other lab members, and 2 industry supervisors, Dr. Tyler Wilson and Richard Michael. Thanks.
Great. We have time for a couple of questions before the break.
Yeah, interesting. Considering your main data set for the transfer is so large, would you considering adding layers that require lots of data, which you can then transfer, such as attention layers or something like that? Or
Yeah, that's a great question. Adding attention layers is an interesting one because intuitively I want that to work, and I've been working away. So this model was published last fall, and since then I've been working on trying to integrate attention layers into the model, but it's been a little bit difficult. So it may be something to keep people posted on, but it's a hopeful, fingers crossed thing.